Good morning. My name is Francesca Girolami and I work in Careggi hospital. And for the first, I want to say thank you to Symposium Director to invite me for this presentation. Next for the slide to go on. Okay. Okay. Okay, since 1970, the principal technique used for DNA sequencing was Sanger method, and this remains the gold standard for sequencing accuracy today. But the current generation of automated high-throughput Sanger sequencing machine read about 2 million bases of sequence per day, and this throughput is very low if we compare with the size, for example, of a human genome, about 3 billion bases per. But as we know, Sanger method was used to sequence the first human genome. This was a very expensive, very long time-consuming project. And in 2001, as we know, we have the draft version of the human genome sequence published. Since 2008, a new technology called New Generation Sequencing Technologies parallelize the sequencing process, thereby massively increasing throughput. So up to about 50 billion bases of sequence per day at reduced cost per base could be possible to sequencing. So next generation sequence approach show great promise for improving the diagnostic yield in genetic and clinical heterogeneous disease, such as cardiomyopathies. And so the question is, what are the implications for the so-called next-generation cardiologists? For the first, we have new opportunities in diagnosis of Mendelian disease, such as, for example, long QT syndrome or hypertrophic cardiomyopathy or dilated cardiomyopathy. As we know, there are a lot of genes involved, and many variant different genes could contribute to the heterogeneous phenotype. But in perspective, next-generation sequence approach could be improved prenatal diagnosis. For example, in non-invasive prenatal diagnosis, for the new research about the identification and the possibility to analyze DNA fetal cell from maternal blood. Then a new opportunity, the possibility to perform molecular autopsy in sudden death victims. As we know, about 35% of autopsy remain with an unexplained cause of death, and the knowledge of the gene involved could be very important for relatives of the victims. Then, for the future, it's very important the next generation sequencing could be applied also for the diagnosis of the so-called genetic disease in general, such as sporadic genetic disease. I mean by whole genome approach. So, by using next generation sequencing for the discovery of new gene involved also in sporadic case. But these new technologies, as we have just talked yesterday and this morning in this meeting, open new challenges, in particular the large volume of data. And so this is a technical challenges for clinical laboratory, but involve also clinical scientists and cardiologists. So clinical laboratories face a major challenge as a consequence of this high throughput, such as optimize the processing and the storage of a large volume of data and analyze so the result. And this is a challenge also for cardiologists because the analysis of large volume of data require a team of geneticists, computer scientists, statisticians, clinical scientists, and cardiologists to work in a team to work together. So a multidisciplinary approach to the data analysis. Then the major challenges are next-generation sequencing costs that are relatively low if we compared by Sanger sequencing. So if analysis of whole genome is expensive, from $6,000 to $10,000, and at the moment used only research, the exome approach costs are very low, about $1,000 to $2,000. And for diagnostic research, as we have just seen from Lopez's presentation, is very important. Could be used a target resequencing of a panel of a gene, for example, 8 to 10 genes, and then we can tagging more and more patients, about 100 patients in only one run, in only one experiment. And so the costs are very low, about $250 to $400, and so it is very important for diagnostic routine screening. But I think that next generation sequencing caused a paradigm shift, a different approach to analyze the data result. So for Sanger method, we have to analyze a single mutation in a candidate gene in a panel of genes. By next generation analysis, you have to study thousands of variants in multiple genes. This is a revolution, such as from Copernicus to Galileo and so on. So the first important question, and during this meeting we have discussed about this problem. We just discussed is the discriminating the disease cause variants. So we have a large amount of variants you have to associate to the disease. And for this is important the bioinformatics, the searching database, and the know-how, obviously. But according to me, the very crucial point is to have a sufficient large family to test co-segregation. And this is not always is possible to have a large family and more than one patient affected in the same family. And to test co-segregation is more complicated when you have many inherited cardiac conditions that are not completely penetrant, such as ACM, or when you have different variants, several genes that may add together to cause disease or modify expressivity. Then next generation sequencing open new ethical concept, and my chairman Sharon have just explained this concept, but I think it's very important concept that genetic testing could produce unexpected information that may be not directly relevant to the original question. So one example, exome sequencing led to the possibility of incidental finds such as a mutation in a cancer-causing gene during investigation for ACM. And so the question is, should be communicated to the patient this incidental find information or not? In Italy, we have a position statement that is the general authorization for the processing of genetic data. And it says, genetic test, screening result, and the research find that entire factual that benefit may be communicated to such individuals if they so request, or if the result find in question are indispensable to prevent those individual health. The question is already open. This is a very crucial point for discussion. Now our result. In Florence, we perform genetic screening for diagnostic use of the A-myosarcomeric gene involved in ACM since 2000. And current diagnostic evaluation, as we know, proceeds by sequencing a large number of genes based merely on the relative frequency of a mutation. We have investigated for the A-sarcomeric gene 372 patients in the space with a diagnosis of ACM. All patients was offered genetic counseling by a multidisciplinary approach that is pre-test and post-test genetic counseling together with geneticists, cardiologists, and sometimes psychologist support. Our result shows that we have a gene prevalence about 64% of positive. So in more than 35% of patients, the pathogenic mutation is unknown, even after very extensive and expensive molecular testing. So in the recent year, we have a different paper about the application of next generation technology in cardiomyopathies. And in particular, we have this very nice paper about the possibility of analyze the giant gene of titin by next generation sequence. As yesterday was just explained this concept, titin gene analysis was not possible by Sanger method, and so next generation sequencing allowed us to amplify our knowledge about this new gene. And so we decided to perform a pilot study 2 years ago. The goal of our first study was to evaluate the capacity of a target sequence to classify, identify mutation candidate gene for Exome. This work in collaboration with the genetic unit of Bergamo and Genoa Hospital. And so we designed a cluster enrichment system covering 36 genes previously described in literature associated to Exome, and we test the first 6 Exome patients negative to standard Sanger sequencing of a Hayes sarcomeric gene. And this is the result of the bioinformatic analysis. We select about 3 variants, so-called high priority, particular convinces for disease causing. And in particular, I want to show us the sample number 6. That he was an old H2 man old, excuse me, with which belonged to a large Tuscany family, with a variable phenotype, with a variable mix of early onset of supraventricular arrhythmia, with other fibrillation, with mild asymmetric left ventricular hypertrophy localized to the apex with left ventricular non-compaction, and restrictive evolution. And by next generation sequencing, we found a novel mutation, alpha-actin 2, responsible, according to us, to Exome and his family. In particular, the co-segregation was tested in 17 family members. And it's important to stress the fact that in literature there is another large family with a mutation, the alpha-actin 2, previously described by Sensorian group in Sydney, and heterogeneous clinical features of our family are reported also in this Australian family. Then we decided to perform during this year a whole exome study, and so we analyzed 12 patients with unavailable and conclusive standard mutation analysis affected by different forms of cardiomyopathies. And these are data filtering results. This is a very stringent, for the first approach, we use a very stringent filter condition. We have found about 50,000 variants for patients, and then we have a filter for function, for frequency, and for conservation. Then we matched with the database single nucleotide polymorphism, and so remained about 95 variants for patients. Each variant was searched in human genetic molecular database and NCBI gene database, and so we select about 3 rate variants for patients, particular convincing to disease causing, so-called high priority variants. This is the data, and you can see the patient, the diagnosis, and the gene contained the rate variant. And I want to tell you about this patient, this family. This is a woman, the proband, this is a woman of 37 years with arrhythmogenic right ventricular cardiomyopathy and planopantal keratoderma. She has a cardiac arrest when she was running in Cascine Park in Florence. And from family history, you can see that the mother had a mild form of arrhythmogenic right ventricular. Then she said that the brother died for cardiac arrest 10 years before at the age of 28 years. And he had a suspicion on right ventricular cardiomyopathy with scalp dermatitis. From next generation exome analysis, we found the desmoplakin gene variant, a novel desmoplakin gene variant, which co-aggregate in the family. We have found the variant in the mother and not in the father. And a recent paper of my chairman, Charro, described this a man with a similar phenotype with a desmoplakin variant. And it's important to stress that it's the first evidence of autosomal dominant form of an axon syndrome. And also our pedigree, you can see, is report an autosomal form transmission of inheritance. But for the conclusion, this is not all that glitters. For example, this is in our family with a high-penetrant hypertrophic cardiomyopathy when the proband have an apical SCM, but the sister have early onset of the atrial fibrillation without no left ventricular hypertrophy. And from next exome sequence, we have selected an high priority according to Haas variant that is a deletion in titin gene. But when we have tested the co-segregation, no co-segregation was found. The other affected relatives have not this deletion. And so, my conclusion, the perspective, next generation sequencing represents the ultimate revolution for mutational screening and is bound to change our practice in the near future. But however, the wealth of information derived from next generation sequencing is not yet paralleled by the clinician knowledge needed to gauge the value of each identified mutation. And so, as for each prior revolution in genetic diagnosis, the unreliable key to understand it is represented by good knowledge of the probants and their family member. So the knowledge, I want to say thank you to my colleagues in hospital, in Careggi Hospital, to my cardiologists, in particular Jacopo Livotto and Franco Cecchi, of Referral Center of Myocardial Disease in Florence, and the other people we take part to this work. Thank you to all.
This stage. So I was wondering, when do you think we should start performing genotyping in our patients? Should we do it in probands, or should we do it when we've collected the family and see how many affecteds we have? What would your approach be?
Our approach began to analyze. We began to analyze the proband. So the first people of the family will arrive to the cardiologist. So for us, probably the most severe cases of the family with the disease. Then if we found the mutation, because not always possible to find the mutation, we communicate the result in our post-test genetic counseling, where this is a multidisciplinary counseling, because there are the geneticists, the cardiologists, and if necessary, the support of psychologists. And in this counseling, we inform the patients of the possibility to tell their relatives about the possibility to perform the test. And so we have a multi-step approach in different time, because it's very important to, as Sharon has just said, and your very nice presentation, is very important to the approach of the patient could be step by step, and not only in only one counseling, because you have to reflect about the possibility to make the test, and the psychological aspect is very important. So I think that to take time is very important.
Yeah, I was just wondering, in context of Dr. Lopez as well, that you identify so many different variants, and it's difficult to understand what is going on in the family. So maybe it would be advisable, and I would like your comment as well, Dr. Lopez, maybe on this, because you were cut off a little bit. But in terms of the segregation analysis, I find it quite important to sort of collect the family and see how many affected before you may consider to do the genetic investigations.
Yeah, absolutely. And even more so in whole exome strategies, more than targeted. The main practical challenge is that we don't have, or we have a very small number of large families, such as the ones that you showed from the one that you showed in the paper, that would allow us to do that. So if we wait for sufficient relatives affected to go on with genetic testing, it might be quite bottlenecking kind of approach.
Yeah, but then you might be very conservative and say, why do the genetic testing if you only find one affected individual in the family?
Yes, true.
Barry?
In routine clinical genetic practice, if you identify a mutation that you think is pathogenic, you then cascade that to the family without necessarily phenotyping the family.
No, our approach is together with cardiologists.
Absolutely. So that's not standard clinical genetics practice, and I think it's your point really, in that in order to be able to detect these complex phenotypes or the possibility of a second or a third mutation, you have to perform clinical phenotyping in parallel with the cascade genetic test, because otherwise you make an assumption about the pathogenicity which may or may not be correct.
So could that be one of the lessons of next generation sequencing in terms of the number of variants we're going to identify would necessitate that we are actually back to square one? We need to do the clinical job ahead of the genetic testing and see how many affecteds we have, because the interpretation is going to be extremely difficult if we do not have a sufficient number of...
Yeah.
Okay.
Francesca, I was intrigued by the titin findings that you had in your cohort with hypertrophic cardiomyopathy, because in the work of Dan Herman in New England Journal, the group of mainly the Mayo Clinic and the Harvard group screened a pretty large group of hypertrophic cardiomyopathy, and they found a disappointingly low proportion of patients with possible mutation. So what is your experience?
By exome analysis, we found a lot of missense titin mutation, titin variant. And for this first preliminary approach to data, we decided to not analyze this titin variant, this titin missense variant, because every patient have 2 or 3 or more titin missense variant. So we don't know if one of these variants co-segregate with the disease, but even if you test the co-segregation, probably is not sufficiently to establish the causative, the pathogenic role of this variant. But about our family, we have found a deletion, and so for us it was a particular convincing data. But co-segregation destroyed our first hypothesis of pathogenicity.
Okay. Last question. Use the microphone, please.
It was focused on the truncating mutations. So we don't know about what was found in for the missense and the other mutations. So I think it fits. She said that they only found the missense mutation, and I believe that there will be another paper with the titin missense mutation in HCM.